Welcome to the SB Grid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SB Grid.
Hi, everybody. This is Jason Key from SB Grid at Harvard Medical School. Thanks for joining our webinar today. Just a quick announcement. This is our second to last in the series for the 2024 webinar. Well, at least for the season up until summer. We've got one more in July. July 9th will be Dennis Bruckner from Duca Hexter's group at Harvard. He's going to be talking about match maps, which is an interesting tool for matching up X-ray diffraction maps that are not necessarily from isomorphous crystals. So it's in SB Grid. It's kind of a cool tool. So be sure to catch us for that if you're interested in matching up your electron density maps. And with that, I'd like to introduce Joey Davis. Joey Davis is associate professor at MIT, just across the river here. And Joey's going to be telling us today about TomoDragon, which takes CryoDragon, this sort of single particle deep learning application that took single particle cryo-EM heterogeneity and moved it to cryo-ET. So looking at visualizing molecular machines in motion through cryo-ET and deep learning. So whenever you're ready.
Yeah, that's a great intro. It's more or less what I was going to say. I was going to talk a little bit about how TomoDragon is sort of a spin-off from CryoDragon. And then to sort of frame the talk, I was going to do a couple things. One, I just wanted to sort of highlight the types of complexes we're interested in and what sort of motivates this work. And I think that that frames a little bit about how we think about structural heterogeneity, both in single particle and ET work. And then I'll spend a little bit of time actually talking about CryoDragon. And in part there, I want to motivate both how the tool works. I think just building intuition for CryoDragon sort of helps us understand how TomoDragon works. And then also how we use these generative models to try to understand biological systems. And it turns out that many of the downstream analysis tools, or virtually all the downstream analysis tools that we've built out to look at CryoDragon structural ensembles, those are directly portable to TomoDragon. And then I'll spend some time going through TomoDragon, how we developed it, and how we're using it. Okay, sorry, I'm gonna get rid of these meeting controls so I can see my slides. Video panel. Okay, great. So, the way that I sort of got into this was sort of a long-standing appreciation that structural approaches, specifically X-ray crystallography and cryo-EM, have been hugely impactful in helping us understand how large macromolecular complexes carry out their sort of structural functions or cellular functions. And the notion here is that you can get a relatively homogeneous complex purified in its active state. You can image it and then use that to build hypotheses about how this machine might work. At least when I was starting, that was less true in thinking about the assembly process. And here I mean, try to understand how cells can take a pile of individual components, properly unfold and orient them relative to one another to give rise to a functional machine. And then if the cell state changes or that machine becomes damaged, we'd also like to understand how the cell could potentially take those things apart and recycle them back to the constituent parts. Right, and so you can sort of define the pile of components pretty nicely through genetics or sort of IP mass spec like experiments. And again, sort of traditional structural approaches allow us to look at these fully assembled machines. And the question was, are there structural tools that could kind of open this black box up between them? And if you could sort of look into this black box, it would afford you the opportunity to really understand how some key disease predisposing mutations actually give rise to those diseases. Many of these have been mapped to the assembly processes or disassembly processes, but exactly how those work wasn't known. Okay, so why has this been a black box? In part, it's because the assembly intermediates themselves are inherently highly heterogeneous, right? There's a lot of different ways that you could put together these pile of components, and it's very difficult to biochemically isolate discrete defined species. And as a result, when you try to purify these assembly intermediates, you get this sort of structural ensemble with a mixture of components. Okay, so when I was doing my postdoc and then eventually starting my lab, I wasn't the only one, but many people recognize that potentially single particle cryo-EM might allow us to look at these structural ensembles. And that's, of course, because at some level, this is a single molecule approach. By imaging, we can get sort of individual pictures of, tens of thousands to millions of particles. And that affords the opportunity to identify relatively rare, but potentially biologically informative states. Because we are counting these particles, we could quantify their relative abundance of the various structural states. If we looked at the structures in detail, we might be able to discover structural interdependencies between them, and that might help teach us something about the order of assembly. And then finally, we'd like to use what we've learned from these structural ensembles to test hypotheses relating sort of experimental conditions, be that small molecule perturbations or genetic perturbations, to the sets of structures that we see. Of course, this is challenging in part because the individual images are awful. And so what we've always done to generate high resolution reconstructions is effectively average across tens of thousands to hundreds of thousands to even millions of particles. And inherent in this notion of averaging is the underlying assumption that the particles are in fact structurally homogeneous, right? Otherwise, it doesn't make sense to average them. Now, of course, we know that that's not going to always be the case, and people have long recognized that. And sort of a trivial extension one can make to this averaging is to say, well, instead of averaging to infer a single voxel array, I'm going to infer a set of k voxel arrays where these are distinct structural states. They're not actually sharing information across them. Now my task is basically to put particles into one of these two bins, assign poses to them, average them, and generate a 3D structure. Okay, so you can do this, with different levels of K. You can also do this hierarchically. And so you might ask, is this simple discrete classification enough to solve the types of problems we're interested in? And I think when people typically think about the sort of continuous structural heterogeneity analysis tools like CryoDragon, they immediately think about sort of conformational changes as not fitting well into this discrete classification model. And of course, that's true. And I've just sort of highlighted that here. If you imagine that you have a protein complex that can transition between two states, here this domain is up, and here this domain is down. You could imagine parameterizing that rotation along some reaction coordinate. And if it were a highly cooperative transition, where there were underpopulated intermediates, then this would fit really nicely to this K equals 2 discrete classification or a multi-class refinement approach, right? You get these two states. Of course, if this is continuous, then we know that there must be some intermediates here, but maybe they're so lowly populated that you don't need to think about them. Of course, the problem arises as the frequency of these intermediates increases. You're sort of left with a choice of either ignoring them, so you can get two molecularly interpretable structures, or you lump them into one of your states, which is going to degrade the quality of that reconstruction. And of course, the sort of extreme example of this is a completely non-cooperative transition. You can either get two structures, neither of which you can interpret, or you throw away 90% of your data, and you solve two high-resolution structures. And there's a lot of sort of systems where I think this approach of like discarding particles because they're not pushing the sort of resolution of the reconstruction makes sense if you're really interested in sort of atomic details, driving chemistry, for example, involved in some enzymatic reaction. But there's other applications of cryo-EM where you might be trying to infer something about this reaction coordinate, maybe how cooperative or non-cooperative the transition are. And of course, if you throw away all this data, you're going to infer that this is a cooperative transition because you don't see populated intermediates, when in fact, that's just an artifact of the way you've chosen to process the data. Okay, so clearly with conformational changes, you'd like to have a continuous model for them. And you might say, well, Joe, you're interested in assembly. That's inherently a discrete process. You're adding subunits to the assemblage as you're building it up. So why not use discrete classification there? And really the problem there has to do with the combinatorics of how these assembly intermediates grow. So we sort of imagine that this box is an RNA. There's two RNA binding protein binding sites. And if you only have these two sites, that would give rise to 4 states. I imagine that you could handle this with discrete classification without much trouble. If you have 4 binding sites, that gives rise to 16 states, and 6 binding sites gives rise to 64 states. And of course, this sort of grows out of control when you start to think about the complexity of something like even the small subunit of a bacterial ribosome. Okay, and of course, biology is going to help us in some regard here. not all these states are accessible. There's cooperativity in some of the R protein binding events where they come in as pairs. There's enforced sequential nature of assembly in certain instances. But hopefully this illustrates the sort of fundamental mismatch between classification in a discrete form versus the types of problems that we're interested in. Okay, so as I was starting my group, we began to think about new ways we could frame the reconstruction task. And specifically, we began to think about other ways we could use the data. So instead of trying to directly infer the voxel array, we imagined we could try to use that data to infer some mapping function. And this will be a little bit opaque, but hopefully it will come clear in the end. So the idea of this mapping function is that it takes as inputs some notion of a position in Cartesian coordinates and produces the EM density at that position. Okay, and so if you want to get back a 3D structure, if you've learned this function, the way that you'll use it is you'll simply rasterize through the box at each position that you've rasterized, you generate a density, and then you sort of fill out your voxel array and you get back a 3D structure. So this alone doesn't actually help you much. You still get back one density map for one data set, but if you start to think about using your data to learn this function, well, then you can start to think about more complex functions you might want to learn. So you might want to learn a function that's parameterized not just by the position in Cartesian coordinates, but also by a surrogate variable. So maybe it's a discrete class K, and now this looks very similar to multi-class classification or refinement. Need not be discrete. This could be some position on, say, a one-dimensional reaction coordinate. You can think about this continuous variable z, very analogous to what I illustrated for this continuous conformational change. And then, of course, the pie in the sky dream of this is that this function could learn to generate density maps as a function of where you are on some low-dimensional energy landscape. Okay, so the obvious question is, how could you learn such a mapping function given the underlying particle images? And this was really the task that laid out for Ellen Zhang, who was the first graduate student to join my lab. And what resulted from her collaboration with Tristan Bepler and Bonnie Berger's group is what folks now know as CryoDrag. Ellen, of course, has gone on and started her own lab at Princeton, where she's doing incredible work sort of continuing to work on CryoDragon. So I'll quickly go through the architecture here because I think it helps understand TomoDragon as well. So the CryoDragon architecture basically consists of a variational autoencoder. One of those modules is this decoder network here. This is basically the function that I just showed you in a blue box in the prior slides. Excuse me. It's going to take some notion of a position. We call that a positional code. At that positional code, it's going to generate an expected density. And then we'll train this model by simply comparing the image that the decoder produces. We call that a model slice with the input image. The difference between those generates a loss function that's back propagated through the neural network weights, and through rounds of training, the decoder network learns to generate model slices that look like the input images. How do we handle heterogeneity? Well, this variational autoencoder framework has an encoder network upstream. Its job is to map these particle images into a low-dimensional latent space. And just as I showed you in that blue box, we're just going to concatenate what we call latent code with this positional code. And now conditioned on this latent code, the decoder network will produce different density maps. So we published this both as a Nature Methods paper and then this Nature Protocols paper that sort of walks through using CryoDragon. And so the way that you would use it, sorry, I'm going to admit faith more. The way that you would use it is that you would train a CryoDragon model for your data set. You're basically going to pass your data through it round after round after round until the networks have converged. And we have some convergence analysis tools built into CryoDragon to help you know when you're done training. Once those networks are trained, you're going to pass the particles through the networks one final time, and they will get projected into this low-dimensional space. And here I've colored this space based on particle density. And so you can see there are things that look sort of analogous to what you might imagine energy wells looking like, with sort of on the order of a half dozen different states and intermediates that sort of allow for conversion between them. Now, there's a bunch of things you can do with this latent space. You can directly use it as a classification tool. If you don't trust the reconstructions from the neural networks, you can simply use it to isolate sets of particles that then you can take back into your favorite reconstruction suite and do all the sort of traditional things we do with cryo-EM. As I'll show sort of at the end of the talk, we found that CryoDragon and TomoDragon are extremely efficacious filtering tools. And I'm happy to talk about why we think that might be, but we found that it's a great way to sort of filter out bad particle picks directly using the latent space. Of course, you can use the neural networks to generate density maps, and you can do that by sampling discrete states. Notably, you don't need to predetermine how many states you expect to see. You can just sample wherever you like. And you can further use that feature to sort of walk along the data manifold, generating structures as you go, which helps you sort of see continuous trajectories. Okay. So a couple examples of how we've used this. The first one I want to start with is work from Laurel Kinman, a really talented graduate student in my lab who actually just defended her thesis. She collaborated with Jingyu Sun, who is a talented graduate student in Joaquin Ortega's group at McGill University. And the question here was to try to understand how a particular assembly cofactor called KSGA is helping to guide the assembly process. So KSGA has been known for a long time. This is actually the kasugamycin resistance gene. But it was long thought to be involved in sort of impacting translational fidelity. More recently, maybe in the last decade or so, there's been some implications of KSGA having a role in the biogenesis process itself. And so we were interested in trying to understand what role KSGA might be playing in assembly of these structures. Okay, so the experimental design is relatively naive. We're going to grow cells in the absence of KSGA with the idea that that will accumulate ribosomal precursor particles that should be the substrates for KSGA. We'll take that pool of particles and we'll either directly image an untreated control sample, again, this should be the KSGA substrates, or we'll treat them with purified KSGA in vitro. This should be the products of the reaction. And if we compare the structural ensembles between the substrates and the products, that should help us understand the structural transformations catalyzed by KSGA. Okay, so let's look at the KSGA treated sample first. Again, we're going to train this CryoDragon model using this data set. We get this sort of featured latent space. You could sample from the sort of highest density positions there, and you see enormous range of structural diversity. Particles that look more or less fully mature, those with KSGA bound, and then those with large structural elements missing. So sort of motivated this by saying we don't want just a couple structures, we want to look at the full ensemble. And because this is a generative network, we can actually sample hundreds or thousands of density maps throughout this learned latent space, which seems like a pretty good idea until you're now stuck in Chimera with hundreds or thousands of density maps and trying to figure out what to do and how to make sense of all this. And so again, a relatively naive approach here turned out to be quite effective. We said, well, we know what the terminally assembled structure should be. And we know how much density we'll expect to see for every RNA helix or every ribosomal protein at each position. And so we'll just dock an atomic model of the 30S into each one of our density maps. And for each structural element, be it a ribosomal protein or an RNA helix, we'll calculate the fraction of the native density that we actually observe. So if a protein's missing, we'll get low occupancy. If an RNA helix is disordered or in the wrong conformation, we'll get low native occupancy. And Laurel's built this into a sort of an automated tool for folks to do this with on their own system. She calls Maven for model-based analysis of volume ensembles. And so I encourage folks to check that out as well. Visually, how this sort of works. Here I'm just showing two different density maps from this KSGA treated sample. And here we'll focus on helix 15 and helix 44. You can see that they're both present in one map, and only helix 15 is present in the other map. We can scale this occupancy to range from 0 to 1. And now you can apply this across the, say, 500 maps that we looked at here, and on the order of 100 different structural elements, and cluster that heat map to try to find groups of structural elements that are similar in their occupancy across the volume ensemble, and then groups of structures across the volume ensemble that are similar in their occupancy of the structural elements. And so, of course, you can find things like extremely mature structures, extremely immature structures. You can find a dominant class of interconversion where the head is formed, but the platform is absent. You can find a minor class where the platform is formed, but the head is absent. And so looking at this gives rise to this notion that there are parallel pathways by which the mature and immature structure can interconvert. We can look a little bit more deeply at binding interdependencies. So we found KSGA bound to the particle. Interestingly, it was right near this sort of key helix known as helix 44, and they looked like they would sterically occlude one another. If we look at the occupancy of KSGA, plotted as a function of its occupancy of helix 44, you can see that they're mutually exclusive. So whenever helix 44 is present, KSGA is absent, and vice versa. Again, we can apply this programmatically across every pairwise set of elements. And then you see some sort of interesting features. So for example, if we look at S3 and helix 35, you can see that that S3 is only occupied if helix 35 also is, but the contrapositive, or whatever the opposite of that is not necessarily true. And because you see this sort of pattern of dependencies, you can draw that as a dependency arrow where S3 depends on helix 35. Okay, we see other examples where they are completely independent. For example, helix 35 can be occupied independent of the occupancy of helix 23, and the same is true for helix 23. Okay, so this allows you to then build up a network diagram of all of these interdependencies. And what was so exciting about this, at least if you work on the ribosome, is that what sort of falls out of it looks quite analogous to these sort of classic Nomura assembly maps that really depicted the order of binding and the thermodynamic cooperativity and binding of various ribosomal proteins. And so, those are really painstaking experiments that Nomura and colleagues did in sort of the 70s and 80s. And most of those dependencies we just sort of see fall out of our data. And perhaps more excitingly, you can actually find examples where RNA helices are used to link the two protein binding events. And so just as an example, if we trace some of these trajectories, you can see helix 28 showed a dependence or requirement for helix 23 binding, which was required for S11 binding, which was required for S2 binding. So we can really sort of trace out a trajectory of assembly of this particle. Okay, so a couple ways that we've used CryoDragon just to complete the story here biologically. I haven't actually told you what KSGA does. So we're going to go look at the quote-unquote substrates. And long story short, the substrates were grossly more mature than the products, which is a little counterintuitive for an assembly factor. But we sort of thought, well, maybe there was something subtly wrong with where KSGA is binding. Maybe it's recognizing a structural deformation in the particle, and we should go and try to look at that. And so I thought I would use this as a sort of excuse to highlight another tool that we built into CryoDragon now and now into TomoDragon. And that is to do this sort of voxel-wise principal component analysis on the volume ensemble. So what we're going to do is we're going to generate a 3D density map for every one of the particles encoded by CryoDragon. For each one of those density maps, we're going to apply a mask in some region that we're interested in. So here we're going to focus on the mask for helix 44. Then we're simply going to perform principal component analysis on the voxels within this mask to try to identify dominant modes of motion or changes in composition or conformation within that mask along the entire volume ensemble, which is what I say here. Okay, so what do we see? If you look at the first principal component that parameterizes this dominant mode of motion, we're sort of excited to see that there were two conformations of this helix 44, one where the helix is sort of tucked into the body. And it turns out chemical probing data from the late 60s actually predicted that there should be two conformations of helix 44, one of which was active and one of which was inactive. The other side of our principal component, you see this inactive conformation. Okay, so given that we have this now interpretable axis of conformational changes, and I'm calling principal component 1, we can look at the marginal distribution of either the KSGA treated or untreated data sets. And so if we look at the untreated data set, you can see it's bimodal. Roughly 2/3 of the particles are in this inactive conformation, here shown in yellow or orange. And then about 1/3 of the particles are in this active conformation that's actually shown as gray as the background density map here. If we instead look at the KSGA treated samples, you can see that they've pruned away this inactive conformation and enriched for the active conformation. So sort of an exciting result. It really looks like KSGA is acting as sort of a proofreader, analogous to the kinetic proofreading that people see in polymerases or tRNA synthetase, where the inactive conformation can form, that gets recognized by KSGA, and then somehow sort of proofread. And so that led to this sort of integrated model You can imagine precursor particles, and this kinetically partitions between productive helix 44 assembly into an active state. It can do so, but it does so with incomplete fidelity, giving rise to this conformation of inactive helix 44. And in data I didn't have time to show you, we believe that creates a binding site for KSGA. Upon KSGA binding, that will displace helix 44. KSGA is then able to methylate its target RNA. That methylation leads to dissociation of KSGA, and then that affords the particle a new opportunity to assemble helix 44 in this active conformation, thereby increasing the overall fidelity of the assembly pathway. Okay, so this is sort of an example of how we've used CryoDragon and tools downstream of it to try to tease apart a biological pathway. And of course, the real question is, like, how do these get assembled in situ, which is what sort of motivated us to build TomoDragon, this sort of new tool. Okay, so how did these differ? So the fundamental difference is that in vitro, we can just make extremely thin ice with single particles that we can image directly. Of course, in cells, be they fibbed or not, we typically have to collect tilt series data to disambiguate particles along the z-axis. And the way this is done is you'll collect a tilt series. From that tilt series, you'll typically reconstruct a tomogram, use that tomogram to pick out individual particles. And then historically, those were averaged by subtomogram averaging. And more recently, people have been going back to the initial tilt series data, as I'll call it going forward, and using that tilt series data to then perform higher resolution reconstructions of the underlying particles. Okay, so we're going to take this sort of latter approach to TomoDragon, as you'll see in a bit. Okay, so how do we think about the sort of relationship between tomographic data and single particle data? In single particle data, in this CryoDragon framework, you have one particle that gives rise to one image. The encoder produces a single low-dimensional latent for that image, and then you reconstruct a single volume for that one particle. In the tomographic case, of course, we have a tilt series set of data, so there are multiple images that the encoder still needs to project to a single low-dimensional latent, which is the representation of that particle in this low-dimensional space. And so Barrett Powell is another fantastic graduate student who also recently defended his PhD. He really set out to figure out how we could, excuse me, take advantage of tomographic data in this autoencoder-like framework. Okay, so what Barrett did is he basically broke the encoder network into two separate networks. The first one he calls encoder A. He thinks about this as a feature extractor. So it's basically you're going to pass each one of your tilt images into encoder A, and it'll generate this intermediate encoding. These then get concatenated, passed to encoder B, and mapped to a single low-dimensional latent. And then we can pass that to the decoder network, plus this notion of sort of position in the voxel array. And the decoder network actually looks very analogous to CryoDragon. It's going to reconstruct the individual images, the tilt images, pixel by pixel. And of course, we will then generate a loss function by comparing the generated images to the input tilt image. Okay, so this all seems relatively straightforward in practice. You go and try to apply it to data sets that you collect, and you realize that things are not always perfect, right? So what you would love to have is, let's say you did this dose-symmetric tilt scheme. You should have 41 tilt images for every one of your particles, and you go and look at your data set, and there's all sorts of reasons that maybe you have some ideal particles, but many other particles that don't actually have the full set of 41 tilt images. Okay, and those could be because there was an ice obstruction, a reflection came into it, the stage was poorly targeted, or maybe you collected on different days with different tilt schemes. And so this creates a problem for two reasons. One is that you don't want to just throw away particles because they weren't ideal, and you don't want to just throw away tilt images because, particularly for molecules that have strongly preferred orientations, you actually need all the tilt images to be able to see them. Okay, so how does one handle these quote-unquote missing tilts without sacrificing either particles or tilt images? And the idea was, well, we're going through multiple rounds of training, so perhaps in each round of training, we could just randomly sample a subset of our tilt images and pass those to the encoder network. And the advantage of this is that at every round of training, there would be a fixed number of tilts. that number is defined by the smallest number of tilts that you see for any particular particle. And then over multiple rounds of training, because this is a random sampling process, the networks are likely to get to see all of the particle images. Okay. So in this example, if there was, the ideal case was 5 tilt images, the minimal number was 3. Every round of training, we're going to randomly subsample 3 tilt images of the 5 available, pass those to the encoder network. Now there's a fixed dimension of the intermediate encoding that the encoder B can use to project this into the low-dimensional latent space. And the goal is to uniformly sample as much data as possible, given that there will be missing tilts. And so you can ask, how does this actually perform? So we could do this with simulated data sets. And here we're just taking 4 sort of assembly states of the bacterial large ribosomal subunit that my lab just sort of knows well and has become sort of a default test case for us. Following Bextor et al., what we're basically going to do is simulate particle images. We'll randomly change the orientation of these, tilt them according to sort of a standard dosimetric tilt scheme, and then we'll corrupt the images by adding structural noise, CTF, and shot noise. And Barrett has a very nice tool here, CryoSerpent, for any method developers that want to do simulations. This is sort of a trivial way to do these simulations. Okay, so how does this work? So here we've passed these particle images through TomoDragon, and now I've colored each one of the points based on the underlying ground truth label. You can see that they nicely cluster into 4 groups. If we look at the structures, the TomoDragon reconstructed structures nicely recapitulate the ground truth. And of course, because this is simulated data, we have the labels, and we can see that there's a beautiful one-to-one correspondence between the predicted label and the true label. All right, so this is sort of massive compositional heterogeneity. And so one of the questions is, given this data set, what's the impact of this random tilt sampling scheme? And one of the things that was sort of exciting for us, we thought this might happen, but it was nice to actually see it, was that this random tilt sampling scheme actually increases the model robustness to overfitting. So here I'm showing, if you just pass the sequential tilts, this is the cross correlation between the TomoDragon generated maps and the underlying ground truth map, and you can see that it increases through some rounds of training, and then eventually there's overfitting in the model and the cross correlation actually decreases. And this happens to some extent with the random tilts, but you can see that the effect is quite mitigated, and that's likely because the model is less likely to overfit, given that the tilts are coming in random orders. You can also ask, how does this subsetting impact the ability to discriminate sort of heterogeneity? And so we could pass subsets of the tilts through each round of training. You can see down to a surprisingly small number of tilts, maybe 10 tilts or so. You still nicely separate out these 4 classes. And you can look at that a couple different ways. Here, this is the cross correlation between the reconstructed density map and the underlying model as a function of the number of tilts on the y-axis and the number of epochs of training on the x-axis. And you can again see, even down to a relatively small number of tilts, you actually get quite high cross correlations. And so we're thinking about other ways we might be able to use this for sort of cross-validation by passing different subsets of tilts to different models. Okay, one of the other challenges in tomographic data is that not all of your tilt images are of the same quality, and that happens for 2 reasons. One is that you're physically tilting the stage, so the high-angle tilt images inherently have less contrast. The ice is much thicker. And the second is that you're imaging the same area over and over again, and so of course you're accumulating radiation damage that degrades the high-frequency signal available, particularly in the late tilt series data. Okay, so really just taking inspiration from a ton of people's prior work, and specifically Grant and Grigoriev, we incorporated sort of dose filtering, and we weighed the loss function in TomoDragon, taking into account this sort of tilt stage angle and the amount of radiation damage. There's sort of 2 ways that we do that. The first is that we actually only use a subset of each one of the pixels for the decoder. This is great because, you know, at the late tilt image data, all the high frequency information is gone anyway, so there's no reason to spend time on the decoder evaluating that. So we actually just mask the image. I should have said all this is done in sort of Fourier space. We just mask the image to the set of pixels that we expect to still have image given the amount of accumulated dose on them. We could take that from these dose weighting curves from Grant and Grigoriev. Moreover, we can weigh the reconstruction loss according to both the dose that's been applied using this dose weighting curve, and then also the tilt angle of the stage on this tilt weight curve. And the goal there is to downweight contributions of the noisier measurements to the loss function, right? We don't want to use all the information equally. Okay, so what are the effects of these two things? The first you can see is that if you either use dose weighting or masking, you get better reconstructions. Here I'm showing the finest sampling of this box. You can see in the absence of either, you get a lower resolution reconstruction than you do with using either or both. And that also manifests in these map-to-map FSC curves. In purple, that's in the absence of weighting or masking. And the other three curves are the permutations of including masking or weighting. Perhaps more importantly, we calculated the wall time. So this is the wall clock time that it took to achieve these reconstructions. And you can see by simply masking out the pixels that you get a much more efficacious training. It's much faster to get to these reconstructions. Okay, so this was with sort of compositional changes. Can we learn conformational heterogeneity? Here we took a beautiful data set, it's my favorite molecule, ATP synthase, from John Rubinstein's group. And so for those that aren't familiar, right, this is this sort of rotary motor that has this complex rotation. The Rubinstein group solved a series of structures of it. Here, these atomic models are sort of colored from purple to orange as it goes through this rotary motion. So of course we can simulate that as we did before. As tomographic data, and then use TomoDragon to embed this in a low-dimensional space. And you can see that the data manifold, now colored sort of from purple to yellow, actually nicely follows some sort of trajectory. We can sample along that trajectory, and then you get back this sort of beautiful video of this machine in motion. Okay, so we felt like at least with simulated data, we could nicely handle compositional and conformational changes in tomographic data using TomoDragon. And so, of course, the question is, how does this perform on real data sets? And so the first we looked at was a data set from Dmitry Turgenov's lab. Well, I guess it wasn't his lab. Dmitry Turgenov published. This was a purified apoferritin. We actually processed this in C1, so we're not taking advantage, or not explicitly taking advantage of the octahedral symmetry. And the TomoDragon reconstructed map is beautiful. You can see, sort of all the features you'd expect to see. If we went and looked at the latent embeddings for this, the sort of surprising thing was that there were three major groups. We can look at density maps throughout those major groupings. The main one here sort of looks like apoferritin. There's an outgroup here, and if we look at either the density maps or the subtomogram picks, these are clearly just false positive picks from the particle picking. And then there was this outgroup sort of shown in orange here that seemingly looks like apoferritin, but the model is clearly seeing something different about it. And that becomes obvious when you slice away, and you can see that this was actually holoferritin, and it constitutes maybe 2% of the data, but it's like beautifully discriminated by TomoDragon and helps to pull it out. Presumably this got loaded in cells during the purification, despite presumably no one intending for this to be a mixture of holoferritin and apoferritin. Looking again at a data set that Dmitry had previously analyzed from Yulia Mohammed's group. This is in pneumonia. This is this amazing bacteria that is so small we don't have to fib it. And so you can basically just look directly for ribosomes in cells imaged by cryo-ET. And for folks that are doing methods development, this is a wonderful data set to work with. Okay, so we're going to pick ribosomes out of it. If we perform sort of traditional subtomogram averaging using warp and M, we get back to a roughly 3.5 angstrom structure. And we were very excited to see that the TomoDragon reconstructions recapitulate those features at similar resolution shells as you see with the traditional tools. And again, like, this is such a cool data set in part because if you go and look carefully at the structure, you can find things like, say, density for chloramphenicol. These cells were treated with chloramphenicol. And so, you're imaging a small molecule drug bound to its pharmacological target directly in cells. And I think it's just incredible both that the traditional tools and things like TomoDragon are able to see this sort of density. Okay, what sort of structural heterogeneity do we see? If you look at the latent space, there's obviously a couple major groups. One of them is this isolated large subunit. And so this is free 50S's that are in the cell. And then sort of this ghost white, I'm showing you where you'd expect a 30S to be. Clearly it's absent. If we look at the sort of heart of the distribution, we find particles that are in different states of translation, either with or without EFTU. And we can find coupled relationships between the absence of TFTU and motions of, say, this helix 17. We can go look at translation states. And so I'm going to just show a quick movie here. Here looking at a 70S particle, we'll splice away to look at the peptidyl transferase center. We can see states with the P site tRNA, those with EFTU and an A site tRNA, and then those with an A site in the absence of EFTU, A and P site in the absence of EFTU. Okay, so we can look at these different translation states. And of course, if you wanted to dig into this, you can quantify the relative abundance of all the translation states that you've seen, of which I've shown you a subset. What other sorts of things can we see? So we looked at some of these reconstructions and found sort of extra density in the periphery. That was curious, but the great thing about ET data is you can just grow the box and see what is in that neighborhood within the cell. We train a new TomoDragon model on this larger box. Again, it's sort of a featured latent space. If we look at some of the particles from that latent space, we can see something that looks quite like membrane. And so this was kind of cool. You can map these particles back, and you can find that these particles here shown in red all map back to the cell periphery. And so this really looks like this is co-translational translocation. In retrospect, it's obvious that this should have been here, but it's kind of one of the cool things about cryo-ET data. You don't necessarily know what you're looking for, but you just look and you're going to see interesting features. When we looked more closely at these, we could see that there were two groups, one of which actually appeared to have extracellular density. We were curious what that might be. And I would say at this point, we're getting sort of particle limited. I think we're down to, 380 particles. But from that, you can generate a 20 or 30 angstrom resolution reconstruction. And, obviously have to be a little bit careful with this, but you can dock in, say, a 70S, components of the sec translocon, and sort of build, an extremely low resolution model, but a model nonetheless of what this translocon looks like bound to a 70S on the membrane. Okay. What other sorts of things do we see? Of course, we see pairs of ribosomes. We expected those to be disomes and trisomes. And so here's sort of a movie looking at these various disomes. And you can see that there is a stereotyped orientation between the two of them, which you'd expect for these disomes, but there's subtle variation there, presumably a result of the sort of flexible linkage from the mRNA between them. We can go and dig in a little bit deeper. We take one of these volumes and now focus on the tRNA, on the peptidyl transferase centers. You can see these tRNAs bound in either the left or the right ribosome, and you can see bridging density between them, which we expect to be the mRNA that's linking the 5' and 3' ribosomes of this disome. Okay, so at the very beginning, I sort of mentioned that one of the things we're excited about with both CryoDragon and TomoDragon is how rapidly or how efficacious they are at filtering data. And as we had built out TomoDragon, we were really excited to apply this to a ton of different perturbations in the ribosome biogenesis process to try to understand how it worked. But at least historically, it's always felt like it's a huge campaign to do cryo-ET. You're going to be milling for weeks on end, you're going to be collecting data for weeks on end, and then there might be 6 months to years ahead of you of data processing just to get to these reconstructions, even of things like ribosomes. And so we were really keen to see if we could sort of do a mix of applications of TomoDragon to expedite the processing, and then work with talented folks like Shaimal Moscolanti at University of Michigan and his graduate student Tyler Brandt to try to push on the data collection size to see how small of data sets we could use to get sort of usable reconstructions to help understand biology. And so what's come out of that is currently a preprint that I encourage people to take a look at. And what we do is we basically rapidly fib mill a relatively small number of lamella, say 20 lamella in a day, and the next day we collect about 60 tomograms from them. And that's our full data set. This is pretty small by comparison to what most people have collected. And then we'll take that and go through rounds of TomoDragon, RelyOn, and IM, using TomoDragon's ability to effectively filter out poor particle picks to try to push the reconstruction. And we can do this quite quickly, where, as you'll see in just a minute, we can get to sub 6 angstrom structures in less than 2 weeks. Okay, so what does this look like? We'll start with particles that have been pretty heavily downsampled. Here, Nyquist limit is on the order of 15 angstroms. We get back a structure that's Nyquist limited. If we look at the latent space from TomoDragon, there's a set of good particles and a set of bad particles. We can segregate those out, re-extract particles at a larger box size. Now Nyquist is about 5 angstroms, and we have a roughly 6 angstrom reconstruction. You can see that the core elements of the core of this go to Nyquist limit. And then here we can see 70s, 50s, errant picks, and what we've assigned as platinum sputter, nicely separated out. Again, we can take the best particles here, the 70s, and push the reconstruction resolution a little bit further, here hitting 5.8 angstroms. And again, this whole workflow is completed in 10 days, and I'm really excited about this opening up the possibility to now look at large sets of genetic or pharmacological perturbations for the assembly process, and then looking at the distributions of structures that come out of it. Okay, so with that, I want to thank the folks that did this work, and then I'm happy to take some questions. So first, highlighting Ellen Zhang. Again, she did all the development on CryoDragon, has her own group at Princeton now, and is really doing well there, continuing some of the CryoDragon work. And then Laurel Kinman recently graduated from the lab. She did all the work on KSGA that I described. She's currently looking for an independent position, and so if you're hiring, I encourage you to reach out to her. She is fantastic. And then Barrett Powell did all the TomoDragon work, another incredibly talented student in my lab who just finished and is on the market currently. So I encourage you to reach out to both of them if you're looking for talented folks. And then I put some GitHub links in here. Some subset of this, CryoDragon and TomoDragon, are already on SBGrid, and I believe Maven is, but I'm not actually certain. With that, I'm happy to take any questions.
Fantastic. Thank you very much, Joey. Questions? Yeah. We've got questions in the QA as they're coming in. If you'd like to, if you're mic'd and like to raise your or ask your question directly, you can raise your hand, but the raise hand button is I just click there. We've already got 2 in the QA, so I'll just take Jeffrey Willard's first. So Jeffrey says, let's consider the UMAP density plots as an estimate of the energy landscape. How can we get error bars, and what are the dominant sources of error?
Yep, good question. Okay, so like pedagogically, I love using the latent space or 2D representations of it to think about energy landscapes. There are no good guarantees that that should actually correspond to an energy landscape, particularly with a UMAP embedding of it where the underlying algorithm is trying to sort of force to cluster things and focus on local connections versus global distances. Okay, so all those things aside, I would say what we sort of are moving towards now is this on-the-fly sampling of every particle. That then gives you density maps for every particles, and then we like to analyze that volumetric space. And so some of these tools are built out, not all of them are. So I think this is what we'll see coming in the next year or something. But the nice thing about that is that then you can interpret the volumes directly without the sort of convolution of the priors that go into the latent space, which I think are going to completely ruin your ability to interpret it as an energy landscape. Now, your second question about like how do we get error bars on this, I think is a great one. You could imagine doing things like, you know, statistical subsampling of the particles and then doing PCA on those. And so that sort of gives you a notion of like a technical error bar, like a technical replicate of what that looks like. But I don't think it gets to the underlying question of like, if I repeated this experiment, how similar would the distribution be? And short of convincing your PI or your funding source to collect replicates of these data sets, I think it's going to be hard to get there.
Yeah, you showed some estimate of what the, like how many particles you had in that ribosome with the membrane. Yep. And the sort of like, so you're able in some way to back out approximately the portion of particles that contribute to that signal. I think that should be proportional to your signal to noise, I guess.
Yeah, I think the number of
Yeah.
particles in that state, the frequency of them should be proportional to the energy that they have, or the energy of that state, right? Is like more and more frequent particles should be deeper energy wells. I think the question is like, what's the error on that? And that I
Yeah.
don't think you can get to without having some
Well,
notion of errors there, right?
well, there's also the fact that like we've now left single particles and we're participating in a bigger chemical process involving a membrane,
Yep.
additional protein components.
Yep.
Like we've left the energy landscape, right? We're in a different place now in some sense, right? We're interacting with other cellular components that each have their own energy landscapes. And so,
Yeah.
yeah.
Yeah, I mean, I think you're still
Yeah.
looking at like the instantaneous distribution of states that were there as imaged, right? And so, to the extent that that is non-equilibrium, that's non-equilibrium, but it's an instantaneous distribution of that. I mean, certainly if you have a cell that's growing and things, it's not an equilibrium distribution, but I think you still have this sort of instantaneous distribution of the particles,
Sure.
and then they be proportional to the energy that they are occupying.
Yeah, I was just thinking along the lines of what you said in the sense that how replicable would these be across, you know,
Oh, sure. Yeah.
another tomogram from
Yep.
another cell prepared identically on the
Yeah.
same, you know, it's might be a little challenging. So
Yeah, so I think
next,
for things that are abundant, right? So I sort of mentioned, well, you could subsample the volumes, and then calculate sort of a voxel PCA to get some notion of the states, and then count the particles that are in different states. When you do that subsampling, you could do that where you basically say, I have 2,000 cells. I'm going to take this set of 10 cells, this set of 10 cells, this set of 10 cells, and then you could get a notion of like, how does this change cell to cell? The changer with doing it that way is that you may have cells that are in different forms, different places in the cell cycle, and you may look like you get big air bars, but that's because some of them are in growth phase and others in mitosis or whatever, depending on the cell type that you're looking at. Yeah, it's a good question.
But also why tomography is awesome.
Yeah,
right.
We're going to learn things. Like I think you
Yeah.
should treat this as hypothesis generation. You should use it to sort of like look at things, find interesting things, and then design experiments to go test them.
Great. The next question has to deal with at what stage do you go forward with TomoDragon in your workflow? So what kind of successes with standard single tomogram averaging refinement tools, rely on WarpM, would be required before you'd be confident moving forward with TomoDragon for filtering?
Yeah, it's a
and
good.
then,
Okay.
you know, do we already need good reference angles before we can use
Yeah.
TomoDragon to reject bad particles?
Yeah, great questions. Okay, so great questions that a reviewer also asked us. Where are the slides for this? Sorry. I may not be able to find these live. So I would say, you know, you really want to have sort of a to start using TomoDragon, you probably need to have like a what do I want to say here? For ribosomes, I would say you want sort of like a sub 30 angstrom reconstruction. And there, the goal would not be to use this as a single round of TomoDragon where you're trying to actually study heterogeneity, but you'd be using it to just do particle filtering to get rid of bad picks. At least in our hands with the picking tools that we've used, this is still a huge source of problems. There's tons of false positive picks that contaminate the reconstructions. And so we use TomoDragon.
Sorry, just to interrupt. You're still on your last slide if you were intending
Yeah,
to
I was
change.
trying to find it and then I gave up.
Okay, sorry. Okay, sorry. I thought maybe I was not seeing the right thing. Sorry.
Yeah, not sure where. Yeah, I have it in this deck somewhere, but there's too many slides in this deck. So we use it relatively early, and then we use that to do particle filtering, and then we take that new set of particles and do a fresh round of reconstructions. That gives us new angles. And then we'll use TomoDragon again, and we sort of do this iteratively until we no longer see benefits. So I guess the long-winded, that's a long-winded way of saying I would use TomoDragon early for the filtering benefits of it, but I wouldn't analyze the sort of structures in detail until you feel like you're no longer seeing benefits from filtering. And that's the point at which, in our hands, it's typically like sort of sub-nanometer resolution from the consensus reconstruction.
All right. Next question, and I'll just jump to the second half first and then come back to the experimental part. Tutorial materials that are available to learn TomoDragon and model proteins that you might recommend to get familiar with it?
Yeah, so let's see. Reach out to us. We're always happy to work with individuals that are interested. Barrett, actually, as we speak, is giving a workshop at University of Michigan on using TomoDragon as well as a bunch of other tools there. So I would encourage folks that are interested in getting into this to go to workshops. They're offered in the States. I know Europe offers a number of sort of related workshops, and so that's a great place to get started. And then in terms of the bib milling, so this is sort of like, Scheinmühl's bread and butter, but I think there's a couple key things there. One is the way we prepare the cells. Because this is bacteria, we just grow them in batch, pellet them at relatively high density, deposit them on grids, so you have this lawn of bacteria. And then we use pretty low angle milling, like 5 to 7 degree milling. So you can sort of cut through a bunch of cells at a time, get a relatively large lamella. And then from those lamella, on average, you can get 3 or 4 tomograms per lamella, given the size of them, where each one of the lamella is basically packed with cells. In terms of why you're only able to get 2, I don't know. There's a lot of specifics there. It's hard to know what's rate limiting, but
Yeah. All right. Any additional questions? If not, Joey, thank you very much. TomoDragon is I'm always in awe to see these conformational maps. And I know it's not drawing making a kinetic molecular mechanism from static structures is never an easy to do, right? But
correct.
But proteins move, and it's part of what they do. And in order to understand them, we need to understand how that works. So it's really spectacular to see. Thank you
Well
very
said.
much for the great talk. And thank you everyone for attending. We've got one more before summer. Be sure to join us in July for match maps. And thanks again.
Awesome. Thanks so much. Appreciate it.
Take care.